Hey everybody, today we're going to talk about the tyrosine kinase and the ion channel receptors. So tyrosine kinase receptors, these are going to be receptors that actually extend through the cell membrane, and the intracellular part is going to function as a kinase, which is going to transfer an inorganic phosphate group from ATP to a tyrosine on a substrate protein. So that's a mouthful. We're going to take a look at it in a second to kind of better explain it. The mechanism, though, is ligand binding like last time. So this time you're going to have two receptor molecules that aggregate. And then the activation of the tyrosine kinase parts happens in the cytoplasm, and then you have the phosphorylation of tyrosines by ATP. So here's a picture where we can actually look at all of these things. So again, the blue part is going to be the extracellular matrix. You have the cell membrane, and then you have the cytoplasm, which is the yellow. So these are the tyrosine kinase receptor proteins, and right now they are inactive and they are separate. And so you can see that there's a binding site on each that extends outside of the cell membrane. Then you have the aphid helix part that is within the cell membrane, so it's going to be sort of interacting with that hydrophobic region. And then you've got the tyrosines down here along these receptor proteins. And then over here you have some inactive relay proteins. So what happens in this system is the signal molecules are going to bind to the receptor proteins. The receptor proteins will come together. All right, so now they are joined together, and so they are activated. And now you can also see that ATP has come in and has phosphorylated or added an inorganic phosphate to each of the tyrosines on these receptor molecules. And so this system has been activated. Now these relay proteins have become activated as well, and so these relay proteins will then trigger a cellular response. Okay, so you've got the signal molecules that are going to bind, the tyrosine receptors are going to come together. They will become phosphorylated by ATP, so then that's why we see these phosphate groups. So this system is active. You now have activated relay proteins, which will then trigger a cellular response. So the intracellular proteins become activated, and they're the ones that are going to cause that cellular response. This receptor system often activates several different pathways at once, and so these are going to be involved in helping to regulate really complicated functions, things like cell division, where you have lots of things happening within the cell all at once because you've got multiple relay proteins here that can trigger multiple cellular responses. The next thing we're going to talk about are ion channel receptors, and these are going to be protein pores in the membrane that are going to open or close in response to chemical signals, and they are going to either allow in or block out the flow of ions, so like sodium or calcium are two really good examples. Again, activated by a ligand on the extracellular side. So again, we have that signal binding on the outside of the cell, and it's going to cause a change in ion concentration inside the cell. So once that ligand binds, this protein channel will allow ions in, and so once ions are allowed into the cell, it will actually change then the concentration of the ions in the cell. So a really good example is how this is used within the nervous system. So here we go, we have our diagram. So these are the ions up here outside the cell. Within the plasma membrane, we have our ion channel protein. Right now it is closed, and so we have a very low or non-existent concentration in the cytoplasm right now of these ions. Here's our signal molecule. That signal molecule is going to attach with that ion channel protein. When that happens, the ion channel will open up, and now you can see we've got some of these ions moving from the extracellular fluid into the cell, and so now we have a change in the ion concentration, and it's going to be that change in ion concentration that's going to trigger a cellular response. And then when this whole thing is over, then that signal molecule will detach and leave, and then the ion channel closes back up again. All right, so this was a quick one today. In your lecture question, what are the main differences between the G protein linked receptors and the tyrosine kinase receptors? So I want you guys to think about what we talked about in the last lecture and compare it to this tyrosine kinase receptor today. Think about the differences in how it acts. Think about the what kind of things these receptor systems are used for, what kind of signals they'll send throughout the cell. All right, thanks guys.